Chapter 1, Section 15, Woes and the Tree of Life. Now, for a lot of times, ever since, I think even predating Darwin, naturalists, the would-be biologists at the time, tried to figure out how things relate to each other. After Darwin, origin of the species, everything's related somehow. We started to try to come up with ways, what's this relationship? Yes, things came before what we have now, which is the basis of evolution. And what we have now is not going to be the same thing that's always going to be here. There was a stepwise. There's what we got now, and there's what came before, what came before, what came before. And if you go back, all the species we have now, there were fewer, previously. What we have now, some of them weren't there yet. But there were other species that just didn't make it. They died off, so we have no record of them now other than maybe a fossil record. So we've always been trying to figure out the relationship of these. Early on, late 1800s, early 1900s, when they tried to do it with animals and plants, they went by appearance, who has certain abilities, who doesn't. If things have abilities, they kind of look alike, then they must have, evolved from the same, common ancestor, and then those common ancestors had a common ancestor, and that's how they came up with, this idea of tree of life, how things are related. They also tried to do this with microbes, bacteria. Well, bacteria, other than the gram-positive, gram-negative, so it gets to be harder to do. So they started looking at, okay, where are they going to find? What are their ecological niches? What are their biochemical abilities? Do they have appendages, flagella, cilia, fimbria? And they started joining together like with like, different species but under the same genus, different genuses but under the same family, under the same class, order, the whole Linnaean thing. And yes, I know I got them all out of order, so don't crucify me. And they started to come up with this idea. Eukaryotes, Plantae, Protist, Animalia. Here's, you've traced back on the branches and you get common ancestors. And the common ancestors had common ancestors and so on and so forth until you got to the first, true living, eukaryotes. Well, we're finding that a lot of it was bunk, okay? It was based on phenotype. And we're now living in the age of genotype. So they started comparing RNA structures, started comparing ribosomal RNA structures specifically, started comparing gene structures, not so much, a gene only looks like a gene, but which genes are linked together, what are the DNA sequences of the genes, and things like that. So they're able to start showing that the tree of life doesn't look quite like that sketch on the last slide, but it looks more like this. Some early protocell. Offshoots of it became what we now think of as bacteria. Offshoots became, the cyanobacteria or the cyanophora. Some of those early cells, developed and started to have, internal organelles. Became what we think of as the protists. Some of those protists started, instead of being single-cell organisms, started, grouping together, doing things together, and you started to get, the early plants, the fungi, what would become plants, what would become fungi, what would become animalia. Is this one set in stone? Is this one correct? Probably not. Because these things, as more things get published, as we learn more, we see more, and we develop better and better processes, this gets changed. This could have been correct 3 decades ago. Nowadays, they're going to look at it and go, oh, hell no, this doesn't go this way. It goes that way, and this one goes over there. And it doesn't, it just completely bypasses all of that and blah, blah, blah. But we attribute this looking at genetic structures as opposed to the phenotypic thing, to Carl Woese. He was a 20th century scientist. He started looking at RNAs. Started looking at, different nucleic acids, structures that they formed, the ribosomes, and it turns out, the ribozymes, things like that, as a means to try to discern relationships. He looked at them for bacteria and eukaryotes, started to figure out, who came first based on, because the more alterations between certain organisms in not only the ribosomal shape, the ribosomal structure, but also in the ribosomal sequence of nucleic acids, or whatever nucleic acids he was looking at, the sequence, the more differences means chances are they're further apart in a phylogenetic tree. The more similarities, the closer they are, the closer the relationship those two species would have. He looked at bacteria on eukaryotes. He's the guy also who came up with the idea of archaea. Things that are not quite the things that are prokaryotes, because they don't have a nucleus or an internal organelles, but they're not quite bacteria. We'll see in a later chapter the archaea have a cell wall, but it's not made of the same stuff that the bacterial cell walls are. So the bacterial cell walls are mostly going to be, so we're going to see peptidoglycan. Theirs, archaeas, are going to be more of a mycolic acid, more of a waxy, as opposed to the sugar protein that bacteria are. But they're not eukaryotes because they don't have internal organelles, so they're still under the prokaryote umbrella. Remember, the archaeas are the ones that are going to be found in all the strange places. These are going to be the ones that are the real extremophiles. So we started looking at, he's the one who started this whole idea, let's look at nucleic acids, the genotypes. Let's look at genetics as opposed to phenotypes or biochemical processes to sit there and discern relationships. Here's what we now look at. There was some, long ago, there was something. And those, last common ancestor, those protocells probably got spread out, ended up in different environments. Some of those in different environments started to develop abilities, abilities that would later become what we think of as bacterial-like. Other ones ended up in other environments that had abilities that were kind of reminiscent of bacteria, but not really, and they didn't evolve internal structures, internal membranes, so they're not eukaryotes. Well, some of those also continued on, developing abilities, getting internal structures, such as a nucleus, getting internal organisms that would ultimately become the mitochondria, that would ultimately become the chloroplasts, and they became, different things, plants, animals, basically the eukaryotes. Is this one truly correct? No. It's a good approximation of what we think happens, and again, as stuff gets published, this one also will get changed. Remember, at what point the distance between branching points has a significance, not just the spacing because it looks cool, but this longer period here means that this common ancestor had to exist for a long time before it truly became the eukaryotes that we would know of today. This long thing means that the diplomonads that became here through some form of speciation, this continued on unencumbered, since almost the beginning. Yes, different species of diplomonads we have now than what were there, but this one long common, whereas you look here, short for plants, because, short for, Thermochus, not quite as long timed around. But hey, they're here. So, as I just talked about, the original cells, they refer to those as the last universal common ancestor. This would have been the protocell. Chunk of nucleic acid. They're now thinking of probably an RNA strand, because the RNA they think was the original, not DNA. Probably had a ribosome or something similar to a ribosome, so that proteins could be made from what was from the information carried on the chunk of RNA. Going to be some enzymes, because things got to be made. There's got to be some sort of metabolism, and then it's all enclosed in some form of a lipid bubble. Ding, 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 ding. That meets all of our criteria for a living cell. So chances are that was the original, LUCA, last universal common ancestor. Over millions of years, as those things made copies and copies of themselves, and they spread all over, different places, different niches. Some of those places, some of those niched, LUCAs became what we think was bacteria. Others became archaea. Fast forward, what was it, 1 billion years? You get the development over millions and millions of years. Internal membranes, and we've got to see what we think of as eukaryotes. We can go, we could study. We can do comparisons nowadays, because DNA sequencing is actually quite easy nowadays. Quite fast, too. You go through, you take whatever you want to study. Here they're showing this plant. You can buy kits now that will isolate genomic DNA or mitochondrial DNA, or isolate all the RNA, whatever. You can send that off to sequence. You don't even have to isolate just one species nowadays. Back when I was an undergrad, to do DNA sequencing, just a small, short fragment of, a couple hundred nucleotides would take days to do, if you could do it. You had to have special equipment, and you had to be really good in the lab to do it. Would have been about here, early 90s. Then they started to develop better and better sequencing methods. Then the hard part was analyses. Okay, so I've got this long string of A, G, C's, and T's. Only way I can compare it to other things is if I have sequenced 2 different organisms, 2 different species, and then I go nucleotide by nucleotide comparing them. Okay, so hey, late 90s, early 2000s, hey, that's when computers really took off. I don't mean that, oh my God, everyone got a computer now. Oh, look, they're just invented. But the computing power in labs, you didn't have to spend new car money to get a really good computer that can handle crunch numbers. We started to look at new programming languages that can do better, faster comparisons of these, tens of thousands of nucleotides, nucleotides in each sequence. Got faster and faster, better and better, until we're here now, where we can do more faster. Still, it's a little cost prohibitive, just because, if you have an N, it's about $100 or so, a couple $100 per sequence. It's not bad compared to what it used to be, thousands and thousands of dollars and weeks, if not months, of work. When they went and did, in the late 90s, they started doing the Human Genome Project. There they sequenced just one person. That took like a year and millions of dollars. It took Cray computers. For those of you who don't remember, Cray computers were the big cabinet-sized things, that would crunch numbers. And yeah, it turns out, they were great at the time, but my laptop I'm typing this on probably had better computing power than those did. So, see how it goes. So as we're getting better at DNA sequencing, and we're really getting much better at DNA comparisons. So not only are we comparing the AGCT sequence of DNA, AGCU sequences of RNAs, but we're starting to see patterns Okay, hey, look, gene A, gene B, gene C here in bacterial A, but over here, look, in this other bacteria, we have A, B, something in between, something after B, and then, oh, look, another gene or so down, there's C. Okay. Does that mean that they got the three genes also? Are they related? Are they closely related? Are they further apart related? Because there's something in between B and C. So we get to start asking questions like that.